So this is the first stage of DNA replication, in which proteins start to attach to the DNA and begin to separate into two anti-parallel strands. We can see here and here and here. And this then creates a replication bubble. There is a region that is Y-shaped at the end of the replication bubble where the strands of the DNA are being unwound, called the replication fork. And as we can see in the center, there's this helicase. And the helicase is an enzyme that binds and may even remodel nucleic acid or nucleic acid protein. DNA helicases are needed during DNA replication since they separate double-stranded DNA into single strands and allow each strand to be copied. So this is the second step of DNA replication. And as you can see, we still have our helicase, but now we have the DNA polymerase, and that is an enzyme that catalyzes the synthesis of the new DNA by adding nucleotides to the pre-existing DNA strands. The new DNA can only extend in the 5 or 3 direction. The new DNA that is made continuously is referred to as the leading strand, and this would be the leading strand, what I just circled. So this is our third step of DNA replication, and as you can see, we have our helicase, our polymerase, but we have an additional DNA polymerase. This DNA polymerase is for the lagging strand, which refers to when DNA is made up of short fragments that are bonded together. A lagging strand requires a slight delay before it is replicated, and it has to be replicated discontinually in small fragments. Therefore, it is lacking. It is missing some fragments, as we can see right here, and right here. So here we have our replicated DNA with our leading strand and our lagging strand. And as you remember, the lagging strand is made up of short fragments that are bonded together. So there's a process required to fix the lagging strands. So this is our final step of DNA replication, and as we can see, the Ozaki fragments have been fixed. They are all joined together now. So these Ozaki fragments, or the individual fragments, requires RNA priming, which serves as the beginning of an Ozaki fragment. These RNA primers are removed by DNA polymerase at the end of Ozaki fragment polymerization. The complete but still separated Ozaki fragments are then joined into a single strand of DNA via the enzyme DNA ligase.